Hello, biologists. Let's talk about genetics problems. We're going to start with introducing the father of genetics, that is Gregor Mendel. He was a friar, which is a monk, in Germany in the 1800s. And we talk about him because he kept these really detailed journals of his experiments with pea plants, the plants that make green peas. And when Gregor Mendel was studying these pea plants, he would find different traits. He looked actually at 7 different traits, and he would see when he bred these plants that had these traits together, what were their offspring like? So he looked at flower color, and in his flower color, he had either white or purple. The purple flower is dominant, which means that you only need one of the allele for this trait to be expressing this trait. Mendel didn't know about alleles, but he still saw these patterns and he kept really detailed notes about them, which is why we talk about his experiments. They were really good. So in your trait, your gene for flower color, you have the dominant trait, which is purple. And when we talk about dominant traits, we write it as capital letters. So this would get the big P. And then you have the recessive trait, which would be a white flower. So it would get a little case letter, a little p, so not capitalized. For a recessive trait to be expressed, you need to have only recessive alleles. So you need 2 if you're diploid. Plants can be more than diploid. They can be tetraploid or quadruploid, but you would need all recessive alleles to express that trait. What your trait looks like is called your phenotype. So it looks like a purple flower, and the genes your trait is for is your genotype. So that would be big P, little p. That would be your genotype. It's your alleles. Mendel also looked at plant height, whether or not it was tall, which was dominant, or short, seed color, and whether or not it was wrinkly or round. So yellow and round is dominant. Pod color and pod shaped, green and inflated, which means you can't see the seeds through it. Those are dominant. Yellow and constricted, where you can see the shape of the seeds, those are recessive. And then flower position, which is even between branches, that's axial, or at the top of the branch, which is terminal. So Mendel did all these experiments where he would breed plants together and then figure out what their offspring were. So we have a way to do this now where we can look at a trait and then figure out its genotype and its phenotype, and then theoretically assume what its offspring are. And we call that a Punnett square. A Punnett square is used to determine your theoretical offspring based on an individual's genotype and phenotype. So if we have 2 plants, a female plant and a male plant, and we're going to breed them together and produce offspring, we're going to look at the genotype and the phenotype of each to determine what kind of offspring we might have. So here you have your mother plant. Your mother plant is producing yellow seeds. Yellow is the dominant trait. I know that this is the dominant trait because there is at least one capital letter in here. That's the dominant allele. So that's what I'll be seeing. Since this is yellow, we get the capital letter Y. That's our dominant trait. This female plant also has a recessive allele. That little y codes for green seeds, but you need only one or more of the dominant trait for that to be the one that's expressed. So even though this female plant has the possibility of having green seeds, it's covered by the dominant trait. So that's a heterozygous individual, hetero being different, and zygous referring to the types of zygotes they could produce. So since this female has one of each allele, she is heterozygous. If this plant were to go through meiosis, she would produce 4 genetically different daughter cells. And in those genetically different daughter cells, she could put either one of these alleles. So here are the gametes that my female plant produced. Some of those eggs are going to have the dominant allele, and some of those eggs are going to have the recessive allele. Here's my male plant. He is additionally heterozygous, expressing the dominant traits. When this male plant produces plant sperm, which is called pollen, half of the pollen will have the recessive trait, and half of the pollen will have the dominant trait. Those theoretical gametes are what we line up outside of our Punnett square. And then we think about what kind of individual we would have were fertilization to occur. So if this capital Y was fertilized by this capital Y, we would be left with a seed that is yellow and has 2 of the dominant allele. So this is a homozygous dominant seed. This plant would grow into a homozygous dominant plant. So this is the beginning of a new plant. This is your zygote for your new plant that would do mitosis. And later this plant would make its own sperm or egg. So here's my diploid individual, my haploid gametes, and the zygote produced after fertilization. But it's random what sperm fertilizes what egg. So if instead my sperm with the little y fertilized the egg, I would have an offspring that had 1 dominant allele and 1 recessive allele, and that would be a heterozygous plant, still expressing the dominant phenotype. I could alter it where the dominant sperm fertilized the recessive egg, and I have another heterozygote option. So you could get the little y from mom or the little y from dad, but you have a chance of getting it 2 different ways. So my last option that my 2 plants could produce are the two recessive alleles in your gametes could fuse together, and you're left with a homozygous recessive plant. This homozygous recessive plant would be expressing the recessive trait. If you only have recessive alleles, then you can see the trait. It's not hidden by the dominant because there is no dominant there. What's our overall takeaway? If we have 2 heterozygous parents, they could produce either dominant or recessive gametes. And our outcome based on genotype is 1 homozygous dominant, 1 homozygous recessive, and 2 heterozygous offspring. What's our phenotypic outcome? Our phenotypic outcome is 3 yellow seeds and 1 green seed. So let's try it again, and let's look at the flower color that Mendel worked with. So here's a purple flower. Purple is going to be dominant. I'm going to write dominant next to it, but I'm just going to write dom. And then our white flower is recessive. So that tells me the genotype of the white flower already. Because it's showing the recessive allele, I know that this little flower is going to be little p, little p. So here's a tiny p, here's a tiny p. If we look up at the prompt, I also have a clarifier that that's accurate. The white flower is homozygous, and it's white. Homozygous meaning 2 the same type. The purple flower has to have at least 1 dominant allele, so that'll be a big P. But just by looking at the flower, I don't know whether or not it's heterozygous or homozygous dominant. And that's where I need to look at the clues in the question. So the question tells us it's a heterozygous purple flower. So that means it's going to have 1 big P because it's purple. And then the other trait is going to be coded for by a little p. I'm going to drop those down and see what my theoretical offspring might be. So I'm going to have the big P's from the purple flower here. And then I'm going to drop this down, little p's. And I'll do the same thing for the white flowers gametes. So it'll be big P, little p, little p, little p, big P, little p, little p, little p. What is my genotypic outcome? My genotype is 50% heterozygous, 50% homozygous recessive. What is my phenotypic outcome? 50% purple flowers, 50% white flowers. So let's erase those. And let's try it again with a different flower combination. Okay. So now we're going to have two purple flowers. One is heterozygous, one is homozygous dominant. We don't know which is which. It doesn't matter. I'll say that the heterozygous one is this one on the side, and the homozygous one is this one up here. But you could swap. It doesn't matter as long as you have one of each. What would be our offspring genotype and phenotype outcomes? I would take this big P and drop it down to both these boxes. So I'll have big P, big P. Do the same thing for the next one, drop it down, big P, big P. All right. Take this big P and move it into both of the boxes to the right. Oops. Sorry. There we go. So big P for both of those. And now here I have my recessive allele, but it's not being expressed by the parent because it's covered by the dominant allele. And that will be true for your offspring as well. So my outcome is 50% homozygous dominant, 50% heterozygous, and my phenotypic outcome, 100% purple flowers. Erase it, and we'll try one more outcome type. Now create a Punnett square for two heterozygous purple flowers. That tells me that both of them have the big P's because they're both purple. I can see they're expressing the dominant trait. But because they're both heterozygous, I know that they both have a recessive allele that's not being shown. And that means that their offspring have an exciting outcome possible. You'll have 50% heterozygous, 25% homozygous dominant, and 25% homozygous recessive. So you'll have one white flower that came from these two purple flowers. And that's sometimes interesting when we see a trait show up in a family and we're like, where did it come from? It could be because your parents are both heterozygous. And so our phenotypic outcome for this trait would be 75% white or purple flowers, 25% white flowers. And our genotypic outcome has three possibilities: homozygous dominant, heterozygous, or homozygous recessive.